All right, let's jump right into this explainer and unlock the microscopic mysteries of the human genome. Think of it literally as your body's ultimate instruction manual, the complete master blueprint needed to build, operate, and maintain you. This incredibly precious manual is stored safely inside the nucleus of almost every single cell in your body, and today we're going to decode exactly how it works. 3 billion. To really wrap our heads around the sheer scale we're talking about here, imagine a book filled with 3 billion chemical letters. That is exactly what's packed inside that microscopic nucleus. If you were to sit down and type out all of these letters, it would fill about 200 thick books, each 1,000 pages long. And honestly, the packaging is a miracle in itself. If you uncoiled and stretched out the DNA from just one tiny cell, it would be about 6 feet long. If you connected all the DNA from every cell in your entire body end to end, it would stretch to the moon and back several times. So here's our roadmap for today's explainer. We'll be looking at the Human Genome Manual, reading the genetic code, the impact of variation, DNA packaging, and meiosis, and finally, patterns of inheritance. Let's start right off with Section 1, the Human Genome Manual. It's pretty wild to think about, but the entire complexity of human life is written using literally just 4 chemical bases: adenine, thymine, cytosine, and guanine. Instead of a 26-letter alphabet like we're used to, nature just uses 4. And they follow really strict structural rules, pairing up perfectly to form the steps of that famous DNA double helix ladder. Adenine always pairs with thymine, and cytosine always pairs with guanine, no exceptions. Now, these billions of letters have to be highly organized so our cells can actually use them, right? The manual is basically broken down into specific recipes for proteins, and we call these chapters genes. But because the DNA strand is just so incredibly long, it can't just float around loosely. It's neatly divided and tightly packed into 23 pairs of massive volumes called chromosomes. You inherit one set of 23 from your mom and one set of 23 from your dad, giving you 46 in total. Moving right along to Section 2, reading the genetic code. Okay, so this sequence right here is known as the central dogma of biology. It sounds super intense, but it basically just describes the directional pipeline of genetic info. First, we have our DNA, which acts as the permanent vault storing your genotype. Because the vault is so precious, we transcribe a temporary portable copy called RNA. You can kind of think of RNA as a courier carrying a single recipe out of a massive cookbook. Finally, the cell translates that RNA courier into functional proteins. These proteins are the workers that execute cellular functions and ultimately create your phenotype, which is just a fancy word for your physical traits. Now, if you're studying this or taking a genetics quiz anytime soon, you will almost certainly see a trap question trying to confuse you about the directionality of this whole process. The 2 strands of the DNA double helix actually run anti-parallel to each other, a lot like a two-way street. So what is the one golden rule you absolutely must remember about how the cellular machinery reads this street? Always 5' to 3'. That's the crucial point. Cellular machinery, like RNA polymerase, can only build new strands in the 5' to 3' direction. Because the strands are anti-parallel, this means the enzyme actually has to move along and read the template DNA strand in the opposite direction, from 3' to 5'. Remembering this one simple directional rule will seriously save you on countless genetic sequence mapping problems. All right, Section 3, the impact of variation. To really ground this in a real-world phenotype, let's solve a biological mystery that affects a huge portion of the global population. Why is it that some of us completely lose the ability to digest the lactose found in milk as we grow up? Get this, lactose intolerance is actually the normal genetic state for mammals. For most people, a C allele in their genetic code naturally causes the production of the lactase enzyme to drop off after childhood. But in individuals who can comfortably drink milk as adults, there's a tiny sequence variation, a T allele, located in a regulatory enhancer region. This single-letter change creates a perfect high-affinity binding site for a transcription factor called OCT1. OCT1 binds tightly to this variant, keeping the lactase gene turned on indefinitely. It's a perfect example of a regulatory mutation just changing the volume of a gene without changing the protein itself. So a single letter change in a regulatory region just changes the volume. But what if a mutation occurs inside the coding region itself? Well, if a mutation like a frame shift variant occurs, which is where nucleotides are inserted or deleted in a way that shifts the 3-letter reading frame, it is absolutely catastrophic for the gene. It completely scrambles the resulting protein structure from that exact point onward, often introducing a premature stop signal that completely destroys the protein's function. Let's move on to Section 4, DNA packaging and meiosis. Because we literally have 6 feet of DNA shoved into this microscopic space, the cell has to wrap it around these spool-like proteins called histones. And here is another high-yield quiz trap for you. If a cell needs to rapidly transcribe a gene, it must be located in loosely packed euchromatin. The cell physically opens up the chromatin so the RNA polymerase can actually access the DNA. If the DNA is tightly coiled up into heterochromatin, it's totally silent and inaccessible. Your cells actually control your traits, partly by just deciding which genes to pack away and which to leave open. To pass this master manual down to our children, our cells use a highly specialized process called meiosis. It's this brilliant biological dance. The cell replicates its DNA once, but then it undergoes 2 complete rounds of division. This reductional division ensures that the resulting sperm or egg cells are haploid, meaning they contain exactly 23 chromosomes, which is half the normal genetic material. This guarantees that when an egg and sperm combine, the new human has the perfect set of 46. And finally, Section 5, patterns of inheritance. You've probably seen this before, but a simple Punnett square is our absolute best tool for predicting Mendelian disease inheritance. Let's look at an autosomal dominant trait, meaning you only need 1 copy of the variant allele to actually show the trait. If we have a heterozygous affected parent, represented by a capital A, lowercase a, having a child with an unaffected parent, represented by 2 lowercase a's, the math beautifully lays out the exact probabilities of what the offspring will inherit. Because genetics has no memory, the chance of inheriting that dominant disease allele is exactly 50%. And here is what's absolutely vital to understand. That 50% probability applies to every single independent pregnancy. Even if this couple already had 3 children who inherited the disease, a 4th child still has the exact same 50-50 coin flip chance, regardless of what happened with previous kids. Now, let's see how this builds, because genetics isn't always perfectly black and white. Even if 2 people inherit the exact same disease allele, it might not look the same. Penetrance refers to whether the trait shows up at all. But variable expressivity, which we see in conditions like Marfan syndrome, means that you can definitely have the disease, but the severity of your symptoms, like how tall you are or the health of your connective tissue, might be completely different from your sibling who has the exact same genetic variant. Let's tackle one last puzzle here. When you're looking at family trees, if a trait overwhelmingly targets males, what does that actually tell us about where the typo in the manual is located? It tells us it's an X-linked recessive trait. The typo is literally located on the X chromosome. This explains so much about inheritance. For instance, if a carrier mother who has 1 normal X and 1 variant X and a normal vision father have daughters, there is a 0% chance those daughters will be colorblind. Why? Because the daughters always inherit the completely normal dominant X chromosome from their father, which protects them from the condition. But for their sons, the story changes completely. Sons only get a Y chromosome from dad, meaning they are totally dependent on whichever X chromosome they get from mom. Because mom is a carrier, that risk jumps to a rapid-fire 50%. If the son gets to mutant X, he will be colorblind for sure because he doesn't have a backup X to save him. So we've traveled from the microscopic 3 billion letters all the way up to how traits echo through generations. With everything we've learned today about how beautifully precise, yet incredibly vulnerable our DNA is to slight variations, it leaves us with a pretty fascinating question to ponder. As our medical technology advances and we learn not just to read the manual, but to rewrite it, if your genome is an instruction manual, who or what will be editing your chapters in the future? Something to think about until next time. Keep exploring, keep learning, and I'll catch you in the next explainer.